In the previous video lecture, we studied in detail the T-independent B-cell activation. It is a two-signal process. First signal is derived from the clustering of B-cell receptors on antigen recognition. And second signal is derived from other molecules of the antigen. We also saw that, as a result of T-independent B-cell activation, mainly IgM antibodies are produced. And, since memory B-cell production requires T-cell help, no memory cells production occurs. Let us now study T-dependent B-cell activation. Recall that protein antigens cannot cross-link multiple B-cell receptors. This is because in these antigens, repetitive and identical epitopes are absent. Thus, when B cells encounter protein antigens, they require T-cell help to get activated. T-dependent B-cell activation is a three-signal process. First signal is generated on antigen recognition by B-cell. Mature naive B-cell recognize and bind specific antigen by its B-cell receptors. Recall that B-cell is also an antigen-presenting cell. Besides recognizing the antigen, they also process them and display them on their surface as MHC2 peptide complex. These B-cells also start expressing co-stimulatory and cytokine receptors on their surface. Most important co-stimulatory receptor is CD40. Meanwhile, the same antigen is also recognized by the mature naive CD4 positive T cells or helper T cells. Note that this recognition by T cell is independent of the B cell. The dendritic cells presents processed antigen in the form of MHC2 peptide complex to this T cell. T cell get activated, and it expresses T cell receptors capable of recognizing these antigens. Thus, at the initial stage of the T-dependent B-cell activation, B-cell recognize antigen and express co-stimulatory molecule CD40 and cytokine receptors. Also, same antigens activate T helper cells. Second signal is derived from the interactions between B and T cell. The B cell and the T helper cell come in close proximity. Recall that B cell display antigens as MHC2 peptide complex on its surface. T helper cells recognize and bind to this MHC2 peptide complex. Once antigen recognition is done by the T helper cell, they now express CD40 ligand on their surface. CD40 receptor present on the B cell recognize and bind CD40 ligand T helper cell. These interactions between T and B cells thus provide the second signal for B cell activation. Third, and the final signal for T-dependent B-cell activation is provided by the cytokines released by the T helper cell. Interaction of B and T cell induce the expression of new cytokine receptors on the surface of the B cell. T cells release cytokines such as interleukin-4. These cytokines bind to the cytokines receptors present on B cells. As a result, B cell starts to proliferate. They differentiate into antibody-secreting plasma cells and memory B cells.